Hey guys, it's me. All right, so hopefully we can get through a lot of this material. Sorry that I'm not going to be there. My car broke down and I can't get it looked at until tomorrow morning. So I'm going to record the lecture for you. If you have any questions, feel free to go ahead and text me or email me. I will answer them as quickly as I possibly can. I'll send you an announcement so you know when I'm on the road. I won't be able to text you when I'm driving, but I will answer as many questions as I possibly can. So let's go ahead and get started. I just wanted to kind of review real quick. Remember, we're talking about adaptive immune defenses. We divided the adaptive immune system into both humoral and cell-mediated systems. The humoral immunity was the B cells, where they start out as naive B cells. That's going to be their inactivated form when they are in the lymph nodes. Once they become activated, they're known as effector cells. They will divide into plasma B cells, which produce antibodies, and of course, memory B cells that will enter into circulation looking for secondary presentation of the same antigen again. Then we have, of course, the T cells. T cells will subdivide into either cytotoxic T cells or T helper cells. The T helper cells being Th1s and Th2s. Those are known as our CD4 T lymphocytes. And then we have our cytotoxic T cells that are known as our CD8 T lymphocytes. There are also some T regulatory cells, or what are called T sub Rs, and those T cells are responsible for regulating T cell activated pathways, turning things on and turning things off. We have also natural killer cells, and natural killer cells play a role in something called antibody dependent cellular cytotoxicity. We will talk more about the ADCC system in a couple of slides. Cell proliferation, remember we discussed about dendritic cells and macrophages actually presenting antigen to helper T cells and cytotoxic T cells in the lymph nodes. So a dendritic cell responds to an injury or an infection. It phagocytizes some bacteria. Because bacteria is foreign and phagocytized, and our dendritic cell, for instance, we're talking about a dendritic cell here, has those pattern recognition receptors. When that bacteria is phagocytized, one or more of those pattern recognition receptors gets bound or gets activated because it is activated by something on that bacterial cell. This will cause the dendritic cell to not only migrate to the lymph nodes for presentation of this antigen, but it will also induce the dendritic cell to produce co-stimulatory molecules. And those molecules are going to activate our helper T cells, which will then divide and carry out their own functions, both activating macrophages, activating B cells, and telling other parts of the immune system what to do. Now, all of this is dependent on the lymphatic system. And so the lymph system is really important, not just for immunological responses, but for monitoring the system at all times. The lymphatic vessels, those actual kind of vessels that mimic, or I call them the back roads to the circulatory system, they hook up with the circulatory system at capillaries. And at the capillaries, fluid from the bloodstream is literally filtered into the lymphatic system itself. And that fluid is called lymph. The lymph will circulate throughout the lymphatic system, moving through the lymph nodes. And the lymph nodes act as the big filters. So the lymph nodes are those secondary lymphoid organs. And in those organs is where antigen presentation takes place. So two different things have to happen at the same time here. We first have a dendritic cell or a macrophage that has phagocytized something foreign, and it will run into the lymphatic system, and it will float around in the lymph until it gets to a lymph node. Once in the lymph node, that dendritic cell or macrophage will have antigen on its surface. It will meet with a T cell and present this antigen to the T cell, and the T cell will become activated. That's one thing that has to happen. The second thing that has to happen for true adaptive immune activation is within that very same lymph, that fluid that was filtered and that the dendritic cell or macrophage was floating around in, within that very same fluid has to be another copy of that same antigen, let's say a bacterial cell. So our dendritic cell phagocytized this bacteria and took it to the lymph node to present, but other bacterial cells from the same population also got filtered on their own. They were not phagocytized, but they got filtered in the lymph. So they also are floating through the lymph and make their way to the lymph node. In the lymph node, this is where they will come into contact with the actual B cell that has its receptor specific for that antigen. Remember, we talked about those B cell receptors and how we're born with these millions of B cells that have receptors on their surface that are specific for millions of different antigens. Well, one of those antigens is going to get filtered in that lymph as well as presented. So the antigen presenting cell has to go to a T cell, but the antigen itself in lymph will be met up by its specific B cell, who will phagocytize it and then present to the T cell. So the T cell gets two forms of presentation. It gets presentation from a macrophage or dendritic cell, and it will then go and look for a B cell that's presenting the same antigen. So in order for a true full-blown memory cell antibody immune response to occur, we have to have the antigen met twice. It has to be met by presentation from a macrophage or dendritic cell, and it has to be met with presentation from a B cell who has phagocytized that antigen in the lymph node itself. All right, so here we have just some illustrations talking about the primary lymphoid organs. We, of course, have the bone marrow, and the bone marrow is where all of our blood cells originate from. B cells will also mature in the bone marrow and then travel in the lymphatic system to the lymph node. So they travel from the bone marrow into the lymph vessels, through the lymph vessels, and house themselves in the lymph nodes. T cells take a little detour first, and T cells have to first go through the lymph system to the thymus. When they are in the thymus, they are going to be primed. Priming is when the T cell is introduced to all possible host antigens. Remember, T cells have to regulate everything. They have to determine what is foreign, what is not, what is a threat, what isn't. T cells are the deciders. They are the ones who have to determine if whatever antigen is being presented or whatever threat there is to the system, the T cells have to determine if it's a foreign threat or if it's a self threat, if the body is reacting against itself. And so T cells must not only know what is foreign, but they must also know what is host, what is part of our own cells. And so that learning process for a T cell goes on. That takes place in the thymus. So in the thymus, the T cells mature, they get primed, they get told and shown everything that is a host component. From the thymus, they then make their way to the lymph node, and then they sit and wait to be activated. That brings us to the concept of an antigen. Now, an antigen is any molecule that induces an immune response. So any molecule that reacts with a B cell receptor, a T cell receptor, antibodies, sometimes natural killer cells, anything that evokes the immune system to respond is considered an antigen, which means we can have foreign antigens and we can have host antigens. If you have a cancer cells give off signals that are not normal. So those abnormal signals are actually considered antigens or antigenic. Now let's break. I want you to look real closely to this small picture up here in the top right where it says epitope and antigen. This is an important concept because when we create antibodies against an antigen, we oftentimes can create multiple antibodies against the same antigen. Now, if we think of a bacterial cell as an antigen, the cell itself, the entire bacterial cell, is not the antigen. The antigen is just some protein that is, or amino acid chain, or sometimes sugars that are located on the outside of the bacterial cell. So in that top right hand picture there, they're showing this yellow squiggly line kind of thing. That is the actual antigen itself that would be attached to the bacterial cell. These are sometimes referred to as antigenic markers. A protein attached to a flagella would be an antigenic marker. The lipopolysaccharide found on the surface of gram-negative bacteria is an antigenic marker. Where on that antigenic marker a antibody actually binds to, what part of that polysaccharide or that lipid or that protein, whatever actual small little component is bound by the antibody, is referred to as the epitope. So you can see in that picture there's this kind of yellow squiggly line here, and then we have a blue antibody attached to one side of that yellow line, and then we have a red antibody attached to the other. So here I have two antibodies that are both attaching to the same antigen because that antigen has two different epitopes. It has two places that we can create antibodies against. And this is significant because we can create multiple antibodies against a single antigen, and we can bombard a bacterial cell. This is very important for neutralization of toxins and metabolites that may be produced by bacteria. This is important for neutralizing and covering up the spikes of viruses so they can't gain entry into our cells. So these antigenic determinants or antigenic markers, that's what these yellow things are will contain multiple epitopes. Not all of them, but many times they will have multiple epitopes, which are the areas of an antigenic determinant that our bodies have been able to create antibodies against. So in the bottom right, you can see in the small picture, there's a large bacterial cell. And on the surface of the bacterial cell, they're just trying to show you where the epitopes are, or these antigenic determinants, and how antibodies are going to bind. So that brings us to the idea of antibodies. Now, antibodies are referred to as immunoglobulins. They are proteins. Most of them are soluble, not all, but many of them are. They are produced by B cells. There are 5 different classes of antibodies, and there are multiple what we call effector functions, or things that occur due to the presence of antibodies. But I want to talk about just kind of your general antibody to begin with. Now, most antibodies are in what we call a monomer form, meaning singular. And because they are proteins, they are made up of polypeptide chains. They are quaternary proteins. They have multiple chains. And I want you to take a look at the bottom diagram here and notice in the different colors there are some regions that are important. So in the dark red, right, the stem of the Y and kind of just above there, in the dark red, this is known as the constant region or heavy chain. The heavy chain makes up the constant region. That's what the FC stands for, is constant region. And for each of the 5 different classes of antibodies, that constant region is going to be the same. For example, I have antibody class G, antibody class A. Antibody class G, all antibodies, no matter what they are specific for, all IgG or antibody Gs are going to have the same constant region or same heavy chain. All antibody As are all going to have the same constant region. It's not the same as a G, but they're all A constant regions or heavy chains. They will bind, notice they're kind of shaped like a Y. The antibodies themselves have 2 binding sites to their antigen. So it's called the ABS, or antigen binding site. You can see the antibody all the way to the right. They've labeled the antigen binding site. Now this is known as the variable region, or VAR, V-A-R region. And it's also called the FAB region. And that's the variable region is made up by the green, blue, and pink These are called light chains, and they are variable. The combinations of amino acids that make up these proteins is going to be different from one antibody to the next. So this is where antibodies become very, very specific to the antigen they're going to bind to. If you have an antibody against the flu virus, that antibody is not going to work against a herpes virus or a hepatitis virus or even another rhinovirus, that it would be like a head cold type of thing. The flu virus antibodies are going to be specific only to that flu and to that strain of flu. All of this gets as specific as the particular strain. The antibodies themselves come from a set of genes that are only found to be active in B cells, which of course is important because B cells are the only ones who produce antibodies. When a B cell is activated, it divides, and one cell becomes a memory cell and the other cell becomes a plasma cell, which is basically just an antibody factory. So in antibodies, during embryonic development, we have a set of genes called V genes or variable genes that combined with the C genes or constant genes, the constant regions of our antibodies, to produce greater than 10 to the 15 different types of antibodies. That is a huge number. We produce millions upon millions upon millions of different antibodies, not to mention how many of each antibody, once it's activated, we actually produce. I like to use the analogy of a million words from the alphabet. We have 26 letters in the English alphabet. Those 26 letters probably make upwards of, say, 300,000 words or 3 million words in the English language. However, the English language is not the only language that uses those 26 letters. There are Portuguese, Spanish, French, other forms of English, Italian, Swedish, all kinds of different languages that all use the same letters. So now we're talking about millions upon millions upon millions of different combinations of the same 26 letters that make up all of these different languages. So we get millions and millions of words from a 26-letter alphabet. The same thing is happening. We get millions and millions and millions of different light chains or different variable antibody binding sites on antigen binding sites on antibodies simply by the recombination, the variable or random recombination of these variable genes. So this is a form of proliferation, and it's sometimes referred to as class switching. So this class switching is these classes of genes are switching around and rearranging and being put together in all of these different random arrangements to give us this 10 to the 15 different types of antibodies. So here we have the 5 different classes of antibodies produced. They're a little out of order on this slide. Normally I would tell you the way that I remember the 5 classes are God-made antibodies: IgG, IgM, IgA, IgD, IgE. G-made, God-made. That's just how I remember it. Now these antibodies, when antibodies bind to something, and I want to make it really clear, antibodies don't do anything but bind. They just attach. That's it. They attach at their antigen binding sites. Their constant regions are going to be sticking out to the environment, and all they do is stick to something. So they stick to whatever it is they are specific for, and that's it. They don't release toxins, they don't drill any holes, they don't chew anything, they don't do anything of the sort. They just attach. But the effect of that attachment is times 6. So we have 6 different things that can happen to an antigen due to the attachment of an antibody to it. The first is the most obvious, right? So we have neutralization. If you have a bacteria producing a toxin, we oftentimes produce antibodies against that toxin. They're called antitoxins. So antitoxins that are specific for a toxin produced by a bacterial cell will attach to that toxin, and it'll get covered by these antibodies, and the antibodies thereby neutralize it. by covering up that toxin, it can no longer gain entry into one of our cells and cause damage. Opsonization, we've discussed. That was an effector function of complement. Opsonization is an increase in phagocytic activity, so phagocytosis is enhanced. The reason for this is, remember in innate immunity how complement had the C3b, and C3b was for binding, and our macrophages and dendritic cells had receptors for C3b on the surface of a bacterial cell. That was opsonization. Well, antibodies work very much the same way. Particularly IgG antibodies are one of our most common. So IgG antibodies have the same constant region. So macrophages, dendritic cells, neutrophils, all phagocytic cells have receptors for the constant regions of antibodies. This way we can make an antibody against anything we want, but as long as the constant region is still an IgG constant region, opsonization is going to occur. So it increases phagocytic activity of other cells. Then we have activation of complement through the classical pathway. We talked about that one very briefly, where C1 protein, remember, cross-links between the two antibodies, activating C2 and 4, which in turn creates that C3 convertase. Immobilization, if we have a bacterial cell that's very motile, has flagella have antigenic determinants, we create antibodies against those. We bog down that bacterial cell. It's no longer motile, it's immobilized. We also have cross-linking. Now, this is carried out by a class of antibody, most often antibody called an IgM. IgM antibodies are really big, and they have 10 different binding sites. We're going to look at a picture of them. And these IgM antibodies can create these really big cross-links between antigens. So we can have one of the antibody binding sites binds one antigen, and another one binds the antigen on another bacterial cell. And eventually this can take soluble toxins and really very, very small antigens and tie them together like a precipitate and cause them to come out of solution if they're toxins, or clot all these cells up. Think about blood clotting. Cross-linking what we call complex congregates is what you did in blood typing. You clump all these red blood cells together so that they no longer look like a nice smooth fluid, but they're just kind of this big lumpy type of mixture. Then we have that antibody-dependent cellular cytotoxicity. I mentioned it earlier when we were talking about natural killer cells. This process is most often carried out by natural killer cells. It is a bridge between the innate and adaptive immune system again. Now here we have antibodies against our own host cells. We have antibodies that are being produced against a cell that is infected by a virus. If that cell is infected by a virus, we need to destroy it. So if we create antibodies against the viral spikes that may be present in the surface of this infected cell, then we will create antibodies against the infected cell. The antibodies will attach. This recruits natural killer cells. Oh, excuse me. And the natural killer cells will come in and attach to those antibodies, release chemical compounds that will then induce phagocytosis. So let's talk about some of our classes of antibodies. We first have the IgMs. IgMs are quite large. We actually have 5 monomer IgMs that are bound together in the middle to create with what's called a J chain. That's the name of the protein. And it creates this big round pentamer structure that has 10 different binding sites all bound together. These antibodies can also exist in their monomer form, and when they're just a single antibody, one of the Ys, that's where we find them on the surface of naive B cells. Remember, I told you in the beginning, our B cell receptors are actually antibodies. Well, they are. They are IgM antibodies in their monomer form. When we put a whole bunch of IgMs together, we get this pentamer form, and in the pentamer form, they're found in circulation. In the monomer form, they're found on the surface of a B cell. They are very large, they can't leave the bloodstream, and they are used in agglutination reactions. So IgM antibodies are what are used when you blood type. We make that's antibody A, those are IgM antibodies. Antibody B, those are IgM antibodies. They are also a significant characteristic of IgMs is that they are the very first antibody produced the very first time you are exposed to a pathogen. That's very important because if you have a patient that comes in and they do an antibody panel, if your IgMs are higher than your IgGs, we haven't talked about those yet, but your IgMs are higher than your IgGs, then you know that this is your patient's first exposure to this pathogen. And you also know that this is early in the infection. This occurs very early in infection in the immune response. This makes sense because the naive B cell receptor is an IgM antibody. So in order to get the ball rolling, the B cells are initially going to produce IgMs. They will later class switch. We mentioned that earlier. We're going to class switch over to IgG antibodies. IgMs are most active in their agglutination reactions, and we see them commonly in blood typing. IgG antibodies make up about anywhere from 80 to 90% of the antibodies found in circulation. They're very small. B cells pump out lots of them. They're the most abundant in the bloodstream. They can also pass into tissue, and they can cross the placental barrier to infants in utero. They are the second antibody produced in primary responses, but the second time you're exposed to that disease, they are the first. And this is because they are found on the surface of memory B cells. So IgMs are on the surface of naive B cells. IgGs are on the surface of memory B cells. And remember, memory B cells are already out in circulation, and memory B cells will not need permission to be activated. IgGs occur in their monomer form only. We won't see them tied together with other antibodies. And again, can cross the placental barrier. This is important so that newborns, when they first come out, they have some antibodies. Their immune systems are still very immature. They can't produce antibodies of their own. So an infant will be born with the same antibody panel as its mother. And they can last upwards of 6 to 8 months in newborns, and they allow time for the immune system of a newborn to mature. IgA antibodies. Now, IgAs are a little tricky because IgAs are the most abundant antibody in the body. We have more IgAs than any other antibody, but they're not usually found in circulation. Most IgA antibodies are going to be found in mucous membranes. They are secreted as a dimer. You can see on the right, the image on the right is two IgA antibodies joined together by their stem regions. And that's where you're going to find them in mucus, tears, saliva, and breast milk. The primary function of an IgA antibody is neutralization. They prevent attachment of antigens to the surface of cells. And again, mucous membranes, gastrointestinal tract, urogenital tract, eyes, respiratory tract, stomach, gastrointestinal tract. They are secreted also by plasma cells that are found in the mucosal associated lymphoid tissue, or MALT. That's where those Peyer's patches are. So we see lots of IgAs in those mucous membranes in order to neutralize any pathogens that we may come into contact with through the mucous membranes. IgE antibodies. Now, IgE antibodies are most often there. We see them in circulation, but majority of them are going to be bound by their constant region to the surface of basophils and mast cells. Now, both basophils and mast cells, as well as eosinophils, but basophils and mast cells are most often associated with allergies and parasitic infections. So IgEs are bound by their constant region, so their antigen binding sites are sticking out. And when an allergen or a pollen, for instance, is an allergen is just an antigen. When it cross-links on a mast cell between two antibodies, remember cross-linking like we saw in the complement system in the classical activation pathway. This is going to attach to these granulocytes. Now remember, our basophils and mast cells are granulocytes. This is going to cause a process to occur called degranulation. In degranulation, those granules get released out into the environment. And these cells store histamines in those granules. So the histamines are released, they flood into the surrounding tissue and bloodstream, and histamines induce a strong inflammatory response. So they cause leukocyte rolling to occur, right? Those selectins pop up so the leukocytes can migrate out. They also initiate vasodilation and fluid leakage and other nerve responses. For instance, you have lots of basophils that are in your nose or mast cells in your nose. And if you are allergic to pollen, not only do those mast cells degranulate, but too many of them and too strong of a histamine response will occur. This will cause a runny nose. It'll cause swelling in the sinuses. It will cause inflammatory responses and, of course, sneezing and eyes watering. Your body's trying to flush it out. Or think about when you take allergy medications, most over-the-counter allergy medications are going to be antihistamines. They're going to be competitive inhibitors of histamine itself. In parasitic functions, all of these histamines and other mediators that get released by these cells are toxic to parasites. So that's why they're oftentimes involved in parasitic reactions as well. Here we have the IgD antibodies. Now, IgD antibodies make up just over 1% of the antibodies found in circulation. They are co-expressed on the surface of mature B cells with an IgM, but not on the surface of effector or memory B cells. They are thought to play a role in activation of these B cells during antigen presentation, but their function in circulation is not really understood. We're not really sure what they're supposed to do. So here's just another slide with some interesting pictures depicting the 6 effector functions of our antibodies. So top left, we can see neutralization. These antibodies attach to toxins and viruses, preventing them from being able to gain entry into our cells. Opsonization, this is that receptor-mediated endocytosis that we've talked about multiple times. On the far right, top right, complement activation using antibodies. So that would be the classical activation pathway. The bottom right, immobilization, preventing adherence. So here looks like the antibodies are binding to flagella, or they could be pili or fimbriae. Then we have in the bottom middle cross-linking, so creating these big aggregates of bacteria so they all get stuck together. This will help with phagocytosis and removal from the system. And then on the bottom left, we have that ADCC reaction where natural killer cells are responding to host cells that are bound by antibodies. Here's clonal selection. Now in clonal selection, we have both B cells and T cells. We get this random reassortment of genes to produce an endless number of cells with receptors to an epitope. This is how we get our B cell receptors. Now, at the bottom of this slide, we have the order in which cells, B cells and T cells, will go through their life cycle. They start out as an immature cell in the bone marrow. They will mature in the bone marrow and travel to the lymph nodes where they become naive. They will remain there as naive B or T cells in the lymph nodes until they become activated by antigen presentation of some kind. Once activated, they then divide and begin carrying out their effect. So they are referred to as effector cells when they are functioning and fighting off some kind of antigen. And then finally, some of them will die off, like plasma cells will eventually die off, and others will become long-term memory cells. So they end up floating around in circulation, and they remember that antigen, and should we ever come into contact with it again, the adaptive immune system will be able to respond much more quickly. Now we have a process known as clonal expansion in B cells. And in B cells, remember, each B cell receptor is specific for an antigen. So at the top of this chart, you can see the hematopoietic stem cell. That's that bone marrow stem cell that's going to divide and become multiple B cells. So these guys divided, and of course, in that first row there called development, we have four different classes or four different B cells, naive B cells. They are in their naive state. And I have each of them is specific for a different antigen, right? B cell W wants antigen W. B cell X wants antigen X. B cell Y and B cell Z want antigens Y and Z respectively. So antigen X gets filtered. It gets filtered through the lymph and comes into contact with the B cell. The B cell phagocytizes it and presents it. At the same time, that same antigen has been phagocytized by a dendritic cell or um, macrophage, excuse me. And that antigen presenting cell brought it and told the T cell, hey, we've got this antigen. And so the T cell found the B cell bound to it, and both antigen presentations occurred, and the T cell activates our B cell. So now we're in this activation state down here in this second row. In this second row, when our B cell gets activated, it begins dividing rapidly. Now it still has IgMs on its surface here, but it begins dividing rapidly, and it moves into the third row here called proliferation and differentiation. In this process here, in this third row, our B cells start dividing into two different types of cells. Some of them are going to become plasma cells, which do nothing but pump out antibodies. That's the only thing they do. Others become memory cells, and those memory cells are going to float around in circulation and determine if that particular antigen shows up again. Plasma cells are going to run around in circulation, and they are just simply antibody producers. They're going to dump tons and tons of antibodies into the system so that those antibodies can carry out their 6 effector functions. They can then go and neutralize or opsonization or activate the ADCC or cause some kind of agglutination or complex congregates. Now, all of this presentation is all well and good, but we have to have our cells have got to be able to present these antigens in a very specific manner. The antigen has to be in the right orientation. We have to know exactly what the antigen complex is. So all cells have some type of major histocompatibility complex. It's known as the MHC. And there are two classes of MHC, a class 1 and a class 2. Now, MHC class 1 is found on all cells. So your liver cell that carries out endocytosis of something, if it gets infected by a virus, it could potentially present that virus on its own MHC, and it likely will, or at least try. So the MHC1 is expressed on all host cells. It is an antigen presenting complex, and it presents antigens that are called endogenous. Endogenous antigens are those that are found inside the cell. Think endo, right? Endo, inside, belong. Endogenous antigens can be viruses, they can be intracellular bacteria, they can be intracellular protozoan or parasitic infections. They can also be cancer antigens or mutation antigens. Something has changed inside of the cell and it's presenting things that are not normal. So that's known as an MHC class 1, the complex that presents endogenous antigens. The MHC class 2 is only found on B cells, dendritic cells, and macrophages. The MHC class 2 activates the adaptive response. It is known as an antigen presenting complex, and it presents antigens that are exogenous, outside. Exo is outside. Think exoskeleton on an insect. So this presents antigens that had to have been phagocytized. The B cell or dendritic cell had to have phagocytized that antigen in order to present it. Now, this starts to get a little confusing, but I want you to remember the CD4 T lymphocytes, T helper cells, they only recognize antigen when it is presented on an MHC class 2. Cytotoxic T cells, they recognize antigen that is presented on an MHC class 1. Okay, class 1. So this helps to differentiate. If you think about it, we need a cytotoxic T cell to respond to a virus infected cell, and a virus is an endogenous antigen. I need a T helper cell to activate, to mobilize or activate antigen presenting cells, or activate B cells, antibody producing cells. I want that to be presented. That's got to make its way to the lymph nodes. I don't want to create antibodies against my own cells, but I want to create it against the antigen. So that brings us to the two different ways that B cells are activated. The first is what's known as T-dependent B cell activation. And in T-dependent, you can see here in box number 1, okay, not the gray one, but the first blue box. Here we have a B cell in the lymph node. It's a naive B cell, and it has been sitting in the lymph node, and lymph has been filtered through the lymph node, and in that lymph was the antigen that this B cell is specific for. The B cell will attach, its receptor will bind that antigen. It will phagocytize, right, and create an endosome with that antigen. It'll degrade that antigen into pieces. Those pieces are then submitted onto the MHC class 2 all over the surface of the B cell, so that this B cell now has antigen, different forms of antigen all along its surface. Now, if this antigen gets presented and it has activated a pattern recognition receptor, co-stimulatory molecules will be produced, and the B cell is essentially saying, hey, T cell, I've got something foreign here. Have you seen this yet? A T cell will recognize the antigen because the T cell's already seen the antigen on a macrophage or dendritic cell. So if if one of these antigen copies on the B cell matches an antigen copy on a macrophage, and both the B cell and the macrophage are producing co-stimulatory molecules, the T cell activates the B cell. The B cell gets permission to then divide and become an antibody-producing plasma cell and an antibody-presenting memory cell. However, if what the B cell is presenting is cell debris or a cell receptor or something that is a host component, because no co-stimulatory molecules are produced, that B cell, even if a T cell recognizes it and activates it, the B cell becomes anergic. It cannot be activated against its own host, against itself. So those co-stimulatory molecules play a really big role in determining whether or not we get memory cells. We do not want memory cells against our own cells. So in T-dependent B cell activation, the memory is the big component here. We get lots of antibodies, and those antibodies get produced, but we also get memory cells. And the co-stimulatory molecule production between all of them is what determines whether or not memory is actually produced. Okay, so now we have T-independent cell activation. In T-independent activation, B cells oftentimes have multiple receptors on their surface. And if we have an antigen that is really huge, really big, then that antigen can have multiple tons of epitopes. An example of a T-independent antigen would be the LPS on gram-negative bacteria. It's very long, it's repetitive. There are lots of epitopes on a single copy of that antigenic determinant. In this case, the B cell kind of gets overwhelmed by this antigen and automatically goes into antibody production. Now, you might think this is a problem because it would induce an autoimmune response, but because there are no co-stimulatory molecules involved in this, the B cell has not phagocytized it, the B cell has not produced any co-stimulatory molecules. There's been no activation of a pattern recognition receptor. We just have this giant antigen that has overwhelmed a B cell. The B cell will temporarily produce antibodies against it and become plasma cells, but there's no memory component. It's a one-shot deal. It'll produce some antibodies against it, it'll respond against it, but there's no memory component. And this is a safety check in the immune system. This is a way for the immune system to say, hey, you might overwhelm me and I'm going to produce antibodies against you, but I'm not going to produce memory cells because I'm not really sure what you really are yet. I'm not sure how dangerous you are. So in this case, the B cell will activate and will produce antibodies against it, but it will not make any memory cells. So there's no memory component in the T-independent activation pathway. So that brings us to our classes of T cells. T cells have their T cell receptors, and the T cell receptor requires an antigen to be presented in order for a T cell to become active. Now we have lots of different T cells. We're just going to talk about a few in here. We first have our T helper cells, and the T helper cells are CD4 T lymphocytes. They will recognize antigen that's being presented by an antigen presenting cell using an MHC class II, so that's macrophages, dendritic cells, and B cells. A T helper cell will, depending on the co-stimulatory molecules that are produced, a T helper cell will divide, and it will become either a T helper 1 or a T helper 2. T helper 1 cells move out into circulation to activate more macrophages. Now, macrophages are already pretty active, but after antigen presentation, a macrophage becomes kind of like a big, giant super macrophage. It becomes super phagocytic. It doesn't present anymore. It simply just goes out and hunts down antigen and just phagocytizes like crazy. T helper 2 cells run around in the lymph nodes and begin activating B cells that are presenting the same antigen. Some will also move into circulation, and they, especially when they become specialized, they become memory cells for those specific antigens, and they will recruit eosinophils and basophils. Eosinophils and basophils produce toxins. T helper 17 cells will also be activated, and they recruit neutrophils. And neutrophils, we know, are highly phagocytic. They kind of clean things up. So these T helper cells are going to eventually, T helper 1s and some T helper 2s, after activation of B cells, are going to move out into circulation, and they go to tissue. And they go to tissue where the site of infection is, or site of injury, that sort of thing. And they release cytokines that recruit other cells to the area to respond. Cytotoxic T cells, known as CD8 T cells, they recognize antigen being presented on an MHC class I. And all they do is find a cell, like a liver cell that's infected by hepatitis. It will present, that infected liver cell will present hepatitis antigen on its MHC class I. A cytotoxic T cell will recognize that MHC class I and recognize it has an antigen bound to it, and it's going to attach. When it attaches to that MHC class I, it will release compounds known as death packages to induce apoptosis. And apoptosis is programmed cell death. So virus-infected cells in particular tend to be very susceptible to the cytotoxic responses like cytotoxic T cells. Cytotoxic T cells are also very important in rejection reactions. So in rejection reactions, we have a lot of that cytotoxic response. Somebody gets a liver transplant. Well, it's not their liver. So the problem isn't that antigen is being presented. The problem is really that the MHC class I on those liver cells does not resemble the MHC class I on the rest of the host cells. And cytotoxic T cells know the difference. So in that case, the MHC class I does not have to present antigen. It's just its presence causes a cytotoxic T cell response. So anti-rejection drugs are trying to turn off or inhibit that cytotoxic T cell response. When liver transplants are made, or kidney, any kind of transplant, when they do the tissue typing to determine if a donor and a recipient match, That matching is the genetic sequence of the MHC class 1. So the MHC class 1, they use a set of genes called HLA genes, and they look to see if those genes match up. And the more closely those genes match, the lower the rejection rate between the host and between the recipient and the donor. That's why identical twins can exchange organs, so to speak. An identical twin, a maternal twin, or excuse me, a paternal twin could donate a kidney to the other paternal twin and there wouldn't be rejection because genetically their MHCs are the same. Now, when T cells get activated, those pattern recognition receptors on antigen presenting cells are really important. Those pattern recognition receptors get activated during phagocytosis. When a dendritic cell presents its antigen to a T cell, that T cell will see those co-stimulatory molecules and know that it's something foreign. So here in this diagram at the top, we have a dendritic cell, and this dendritic cell is phagocytized something. And if we go to the left here, where all these different colors are, you can see that our dendritic cell has both MHC class 1 and MHC class 2s. So it is producing all of these different antigens here. And if it's an MHC class 2, it's going to activate a CD4 T lymphocyte. If it's an MHC class 1, it's going to activate a CD8 T lymphocyte. That's what they're showing you in this diagram here. And of course, on its surface, it's also presenting co-stimulatory molecules. So these T cells become highly activated and they move out and carry out their function. If we go to the right here we have a dendritic cell that's presenting self-antigen, maybe some compound from like cell debris, that sort of thing that got broken down and phagocytized. There's no co-stimulatory molecules here, but it is presenting on both an MHC1 and an MHC2. So here, our CD4 T lymphocyte and CD8 T lymphocyte do both bind the CD4 to an MHC class 2 and the CD8 to an MHC class 1. They do bind, but there's no co-stimulatory molecules. Because there are no co-stimulatory molecules, these two T cells become anergic. They're going to enter into apoptosis. They cannot be activated against anything because if they were, they would be activated against host, and we don't want to do that. So an anergic cell is a cell that's been activated, but it's not allowed to carry out its effector function. So in turn, it enters into apoptosis.
In this video, we can see a T cell that becomes activated when it interacts with a dendritic cell. The T cell is labeled with a dye that fluoresces when it binds calcium ions. At the moment, the T cell is not activated. Its intracellular calcium concentrations are low, and so little green fluorescence is visible. As the T cell contacts the surface of the dendritic cell, we can see it suddenly fluoresce bright green as it becomes activated. However, it still continues to move, crawling over the surface of the dendritic cell, perhaps to sample the cell's display of peptide MHC complexes. Eventually, the T cell loses interest. While it is still contacting the dendritic cell, you can see the fluorescence start to fade. The T cell then migrates away from the dendritic cell.
So in T cell activation, remember, we have these antigen-presenting cells. That's what APC stands for. They're going to present antigen with co-stimulatory molecules. The T cell gets activated, proliferates. In that proliferation, some of those T cells will become memory. Some of those T cells are going to move over and they're going to start activating B cells. The B cells will start making antibodies, and now we have a full-blown immunological response. If an antigen-presenting cell presents without co-stimulatory molecules, the T cells are anergic. Anergic T cells have no response, no activity. They're going to enter into apoptosis, and this most often occurs because a healthy host component got presented instead of a true antigen. Here we have CD8 T cells, or cytotoxic T cells, and in the top here we have our MHC class 1, right? So here is a nucleated cell. This could be a skin cell, this could be a liver cell, kidney cell, cardiac cell, it could be any cell, right? So it's a normal cell. and it starts presenting some proteins on its MHC class 1. To the right of that is a CD8 T cell, and cytotoxic T cells are not going to recognize proteins that are presented on a healthy cell. Okay, so in that top part A there, in that top diagram, we have a healthy cell that's presenting peptides, normal proteins on its surface, and the CD8 T lymphocyte says, yeah, that's totally normal. There's not a problem there. You're fine. And it doesn't do anything. In the bottom, in the green box, part B, here we have a cell that is presenting viral proteins. This could be a viral spike or part of the capsid or maybe part of a protein that the virus brought with it. Regardless, it's presenting a protein that is not recognized by the T cell as normal. And it's going to present those on its MHC class 1, which binds to the CD8 receptor of these T cells, and the T cell begins releasing cytokines. These cytokines are going to induce apoptosis to the cell that the T cell has bound to. So it releases basically toxins that tell the cell you need to go into apoptosis. And this is why it's called a cytotoxic T cell. Cyto being cellular, toxic being toxin, and it's a T cell. So it's a cytotoxic T cell. It is toxic to other cells. Macrophages. Now, initially, phagocytosis is not usually enough to get rid of a particular pathogen. Think about bacteria growing in population, right? There's millions and millions of them. But if we increase the phagocytic activity or the ability to carry out phagocytosis, make the cell more effective or make the cell better at it, then phagocytosis can become a major component of fighting a pathogen. So T helper cells will activate. When a macrophage presents to a T helper cell, part of the cytokines the T helper cell releases is going to tell the macrophage, hey, you don't need to worry about presenting anymore. You just go out into the system and you phagocytize as much of that stuff as you possibly can, as fast as you can. So those T helper cells give them a chemical signal that says, increase your ability to produce lysozymes, get bigger. So the macrophage will literally get much larger, and sometimes its membrane will ruffle up so it has a lot of surface area. It'll increase its phagocytic activity, and the T cell itself, all of these cytokines will cause stimulation of cytotoxic T cells, start recruiting them to areas so that they'll start looking for things like virus infected cells. Now here we have dendritic cells, and dendritic cells are the main antigen presenting cell in the immune system. They do more to present antigen than any other cell. They are super phagocytic, kind of like the supermen of phagocytosis, and there are two different types of dendritic cells. We have DC1s and DC2s. What makes it become a dendritic cell 1 or a dendritic cell 2 all depends on T cells. Now, on the diagram below here, I don't want you to worry about the names of all of these cytokines or compounds or anything. I just want you to see that an immature dendritic cell 1 on the left there will become a mature dendritic cell 1 when it interacts with T helper 1s. And a DC2 will become mature and activated when it interacts with a T helper 2. So DC1s are determined by T helper 1s, and DC2s are determined by T helper 2s. Now here we have the natural killer cells, and natural killer cells are not antigen specific, they are antibody specific. They're responsible for the antibody dependent cellular cytotoxicity response. They recognize antibody bound to self cells, to host cells. So if you have a cancer cell that's emitting these cancer-like antigens, antibodies will be made against it. We're gonna get antibodies against it, and those antibodies will attach. And when they attach to that cancer cell, those natural killer cells will recognize that, and they'll attach to the antibodies and induce apoptosis. So natural killer cells have become a really major component in cancer therapy right now as a form of chemotherapy, immunological chemotherapy. And some therapies are introducing more natural killer cells to a system. Others are giving antibody treatments that are specific to, they're called tumor specific antigens or TSAs, They will give the patient an antibody treatment that targets tumor-specific antigens, tumor cell antigens, which will then induce this ADCC response, antibody-dependent cellular cytotoxicity. It's really a very exciting type of therapy, really exciting time in cancer therapy, because they're starting to be able to use the immune system to correct what it got wrong. Cancer is considered a mistake or an error by the immune system. The immune system missed something, and so these cancer cells were able to grow. So now it's a way to use the immune system to actually treat some of these cancers and use the body's own defenses to correct things. So the adaptive immune response, this is just kind of an overall slide. It just kind of gives you an idea of some of the reactions that occur. You have a copy of this in your outline. So if you don't have the outline, go ahead and print that out. You can see a copy of this where we have antigen presentation and we have B cells making antibodies and all of the different effector functions. I believe this is also something similar to this is located in your textbook. This is another kind of overall slide that's just kind of showing you everything that's happening in an immune response, activation of B cells, activation of T cells, and how they're all interacting together. We have both the humoral response in green there with antibody production and the cell-mediated response on the right with our cytotoxic T cells and apoptosis. Now, I want to talk real quick about some of the different effects and consequences of adaptive immune responses. Now, here we have a table, this kind of graph, and the red line is representative of IgM antibody count, how many IgMs are in the system of an infected individual. And the black line is IgG antibodies. And what I want you to really see here is the very first time from 0 to about 21 days. So this is the very first time at 0 that anyone is ever exposed. This is primary exposure, first exposure. Notice, IgGs are getting produced, but the first antibody to really increase and show up, right, to be that we're able to really detect, the very first one are IgMs. And IgGs do show up a little bit later. You can see that they show up and they are in greater numbers, but we're looking at about a 24 to maybe 36-hour difference. So 24 to 36 hours before IgGs show up on the radar, IgMs are going to be seen. That's initial exposure. But at the end of this stage of illness, at this primary exposure, let's say this was the flu. At the end of the flu, we have memory cells. Remember, all of this activation results in these memory B cells that are going to be out in circulation. Now, memory B cells have IgG antibodies on their surface, not IgMs. So the second time we get exposed to that same flu virus, note how high the IgG antibody production is. It's huge. It's a massive response. We get millions and millions of antibodies produced very, very rapidly so that everything gets neutralized real quickly and we don't get sick. The virus or the antigen, the pathogen cannot take hold and make us sick. In the meantime, we still get, you see this red line, we still get some IgMs produced, which means we're still activating naive B cells. And that's that boosting. We're going to boost the number of memory cells and make them even more so that if we get exposed a third time, the antibody response is even bigger. Here we have this is just IgGs. This blue line is just representative of IgG antibodies. So the first time we produce IgGs, the number's okay, right? It's, we get some produced, we're able to get over the illness. But the second time, because memory cells already have IgGs on their surface and they are already out in circulation, we don't need any kind of antigen presentation. Because of all of that, the IgG numbers skyrocket very rapidly. And this allows for that really quick response by the adaptive immune system to prevent infection. And here's the last slide. In our last slide, I just kind of wanted to show you the effects of disease on the immune system. So here in red, we have what we call a viral load. How much of an HIV virus is actually in the system? And in blue, we have we're looking at the number of antibodies against HIV. And what I want you to notice is the HIV level goes up pretty rapidly, right? These viruses get inside of cells, they kind of hide, and they move up very rapidly. We get a high count, and then all of a sudden, after 1 month, if you notice after the number one here, that's time in months. After 1 month, the number of HIV viruses starts to decline. Now, this is not just eclipse period. Eclipse period occurs very, very early on. What we actually have here is a decline in the viral level because in the blue line, you can see our HIV antibodies are moving up. So at about month 2, we're starting to see our antibody levels increase above the RNA levels or the HIV viral levels. Now, you would think, great, that means that our antibodies are working, our immune system is responding, and it is. But what's the issue here? The issue is that HIV infects the very cells that cause this to happen. So the T cells that would activate B cells and tell everybody what to do are being destroyed by HIV. So even though our HIV levels start to drop, our antibody levels start to level off and they don't continue to increase in order to completely get rid of that HIV virus. And that is because the HIV virus is literally destroying those cells that are fighting against it. So everything starts to level off, and eventually these two are going to crisscross in the opposite direction later on, and we're going to have fewer antibodies than we are HIV. And so HIV would then take over. So this is the gist of the adaptive immune response. I know this is a whole lot to take in, and I'm sure you have a million questions, and I will answer as many of them as I possibly can. If I have time, I am going to try to log in and maybe answer some live questions, do a review with you guys, maybe tomorrow afternoon, tomorrow night. As soon as I know what's going on with my car, I will let you know. I will text you. But I hope that you can listen to this a couple of times, listen to it in short sections, follow along in the book if you can, or with your outline. And again, feel free to email me or text me with any questions. I will hopefully see you guys Tuesday morning. Have a great day. Talk to you soon.